Welcome back, guys. After many days I decided to do some video on the practical purposes of microbiology, because I'm a person of microbiology and I haven't done so much of video on microbiology, and especially these are the basics of microbiology practicals. So let us talk about the practical microbiology, and among them will be, in this first video we'll be talking about just the introduction to that. So practical microbiology, and this is an intro about that practical microbiology. So let's talk about it. Now I'm going to say many important scattered things first in this video, because you need to get started with different instrumentation and different kind of practical, because a lot of practical that we are dealing with in microbiology are obviously dealt with microorganisms, right? So microbiology is obviously dealt with microorganisms, and example mostly of the microorganisms are going to be bacteria in our opinion. Bacteria in our opinion. So once we are dealing with bacteria, now what we need to do, we can't see bacteria with our bare eye, right? So we need to see bacteria using the machine called microscope. We all know that. So we need to use a microscope to see this bacteria. So let's write it down in a table like form. So we need to see, for seeing those microorganisms we need to hold on to microscope, right? And we can't see bacteria even with the microscope without being coloring those bacteria, because they are colorless and they are very thin, as you all know. So you can't really see them. Light can directly pass through that part and you can't visualize them. So visualize them properly, we need to stain them, right? So we need to color them. So coloring is termed as staining. So the second thing we require is coloring them, and coloring those microorganism can only be done using staining, right? This is the second thing that we are going to learn. Microscopy, staining is another thing. So you take this bacteria or whatever microorganism it is, you need to color them using staining processes. Then you need to put them into the slide, because remember you cannot see it bare eye, so you need to prepare slides. So you may require slide. You put the bacteria onto the slide, you stain it. After that you use a microscope to see that bacteria or any other microorganisms like fungi and all these things. So that's the very basic thing, to visualize the microorganism under the microscope. So that's the first general type of process. Now once you visualize them, now this is kind of one side of it, so visualization. Except for that, you need to do many other tasks. For example, sometimes you need to not only see them, but you need to grow them, right? Because to understand them, you need to examine them. So for examining them, you need to grow them, right? So this is for seeing. So visualization, this is for visualization. And if you look at now, we'll look at growing and growth. Now for the growth of the microorganisms, what else you can do? You need to culture them. What we call about growing, we simply term as culturing microorganisms, right? And culturing of microorganisms is nothing different than the growing of any other things, because for the growth what we require food. That's a simple answer. So we require food for growing. So we put food for microorganisms. MO means microorganism here. So we need to put food for microorganisms so that they can grow on it. Now it's very easy to grow microorganism, because what you need to do is simply need to supply some food, and we need to add the microorganism there, a very tiny and little amount of microorganism there, and those tiny amount of microorganisms start to eat that food and start to grow on that food or over that food, right? So it's very easy, just like you've seen many microorganisms growing in front of your eyes, because though they are not micro, you can see with your eye, but they are very small. Like you already know different types of mushrooms, they are growing all the way when monsoon came, and many different conditions when there is a lot of moisture and dead materials. So those type of mushrooms and all those type of fungi start to grow over those dead materials and food, right? So these are the same type, so they can grow on to that food material on the food surface. That's it, right? We need to put a matrix so that they can hold on to it, and second thing, we need to put the food source which contains all the nutrient sources for the growth of microorganism. And it's not again a kind of different thing, because the nutrition that microorganisms require is not very much versatile than human being. They are very basic type of organism, they take very basic type of food materials, and the example of those food material is so we'll be talking about those food, right? So these are so you need to use this culturing process to grow them. So we use microorganism microscopy to visualize them, we use culturing process to grow them. So two of the important things we have done, right? So these are the major two things that you need to know and grow for microorganisms and other things. Now once you have done this thing, now what else you need to do? Sometimes you need to look for the number of microorganisms, you need to count the number of microorganisms, right? So that's also required when once you're dealing with them, right? So it's simply like microorganisms like your pet. If you are a microbiologist and you are going to work a lot with microorganisms, so they are going to be like your pet. You need to see them, you need to take care of them, you need to grow them, you need to apply food for them. So many things are required there. So similarly, sometimes you need to count, right? So to count them and look for them, whether what's the structure and what they are doing actually. You need to count. So counting can be done using microscopy. Two important processes there. It is using microscopy, the simple type, and also you can use a process called uh, what we know as cell sorting machinery. So cell sorting machinery we can also use to know the number of bacteria or any other microorganism cells present in a particular solution, confirmedly or directly. These two are kind of direct processes. These two are kind of cell sorting is also termed as flow cytometry, so you can go for it. These are the high profile techniques, but except for that, the general process of knowing whether there is microorganism present in your medium or culture media, that's what we call it. The food for the microorganisms are called culture media. Remember, they are called culture media. So in the culture media, whether there are microorganisms or not, how you can look for them? To know that, there is a very basic process, and that basic process is simply the colorimetric process. Simply you need to check the OD, right? So optical density check is the simplest way for knowing whether there is bacteria or other microorganism present in the culture media or not, right? So you can look for them by looking at the viscosity of the medium. So these are the things, these are the things that are related, these are the things that are required throughout the place: visualization, growing and counting those microorganisms. Now remember, microorganisms are everywhere. Literally they are everywhere. Wherever you are looking at it, wherever you are touching, everywhere is there are presence of huge number of microorganisms, and they can grow anywhere. Literally, that's what I understand throughout the microbiology career. But the important thing is that for that reason, so suppose you need to grow a particular type of microorganism, so it's the most important task that it should not be contaminated by other microorganisms which are undesired, because literally they are everywhere. So you need to go against contamination, right? So you need to exclude unnecessary ones. You need to pick only those microorganisms you require to work with, right? So that's the most challenging part about those microorganisms and about the microbiology for culturing bacteria and other microorganisms in the lab. So for that reason, what you need to do, you need to sterile certain things before starting this process. You need to sterile each and every materials that is going to contact directly with your culture media or the microorganism, right, or your desired microorganism, right? So you need to sterile those things. So sterilization, I must write here, sterilization is another very important stage for microbiology and any kind of culturing process, whether it's a microbiology culture or cell culture of animal cells and whatever. So sterilization is very much required, and probably one of the most important thing you should take care of before even visualizing growing them, because sterilization comes first. So you need to sterilize them, and for sterilization what we can do, you can use different sterilizing processes. You can use chemical sterilizer, but mostly we use steam sterilizers called autoclave means which are steam sterilizers. Okay, you can use dry heat as a sterilizing approach. You can also use chemicals and other materials, but this autoclaving is the most convenient way to sterilize all of the thing, right? So we will be talking about all of these different techniques like sterilization for microbiology, counting the cells, we'll be talking about growing them, we'll be talking about visualizing them, and all these different parts we'll be talking about in the future videos. So this is all about how to connect these different things, how to work with a particular bacteria or microorganism in your lab, visualizing. So you need to sterilize first all these things, whatever is in direct contact with your culture media or with your bacteria of desire. Then you need to visualize via staining and microscopy. Then you need to grow them in culture media, you need to count them, and then you need to do rest of the job whatever is there. But these are the major basic techniques you must learn as a microbiologist in the very first duration of your course, right? So for these different purposes, different machineries are required, different glasswares are required. For example, for microscopy you require microscope. For staining you definitely require those slides and cover slips, right? And for growing this particular bacteria, you need to require special media called food containing media, and also the matrix. You need to provide the solid matrix, and the matrix here always work like agar. We'll be talking about them later in detail. And we need to have a glassware like petri dishes, because it is having large surface area so that the bacteria can easily grow, because they usually grow onto the top of their food matrix, right? And for counting them also you require microscope, and for OD checking you require different colorimeters. So many machines are required for this process. If you are a microbiologist, you start doing your lab, your practical, your lab work, you start to see many different basic machines that should be there. And obviously for growth, I missed a point that for growing those microorganisms, not only food source is important, but also temperature plays a vital role. And for the temperature, we need to manage the temperature, because they usually grow at not at room temperature, actually at physical temperature, like 37 degrees Celsius temperature to 48 degrees Celsius temperature to 45 degrees Celsius temperature for 24 hours to 48 hours, depending upon the type of bacteria and other microorganisms you are dealing with. So for that type of heating you require a heater or incubator. So incubator is another very important machine for these microbiologists. And you should not these are the very basic things. You should require all of these things for the process. And also after sterilization, what you need to do, you need to culture. For the culturing process you require another machine where you must have done all those transferring tasks, because air is filled with microorganisms. So if you are doing it in your simple working desk, open air, it will catch huge amount of contamination. So sterilization won't play a vital role in that case. So what you need to do, you need to do this in a sterilized room or in a sterilized region where there is no microorganisms, or which is microorganism free. For that we need to use separate type of cabinet that is called as laminar airflow machine, or LAF. We also say laminar airflow or LAF machine. So these are the very basic things. You must have an incubator, a lab, a culturing media. You must have your desirable bacterial culture, should have autoclave in your lab, a microscope, stains. So these are the very primary thing, most primary thing about all the basic microbiology tasks and works that you need to start in the lab. So these are the things, and now in the future video we'll be talking about each of those sections in detail, and we'll be talking about them.